Hello everyone. I'd like to go through some key points from chapter 14 with you. This will be definitely an abridged version of chapter 14. We'll go through what I feel are the most important points, and that will be sufficient for us for Biology 1. So you can base your studying off of what's in this set of slides, this video, and that'll help you to narrow down your focusing. So let's take a look. Chapter 14 is about biological diversity, and let's just start with a definition. So we need to be, in this chapter, we're going to be thinking about how does one species perhaps branch off into multiple species, and so we should start just with a basic definition. What do we mean when we talk about a species? So a species, the most straightforward definition that we could probably have for a species is a group of individuals who are capable of interbreeding and having offspring that are fertile. So this is describing a line that can continue for many, many generations. So that's how we'll define species, a group of individuals that can interbreed and produce fertile offspring. Okay, so thinking about a species, let's start by considering the question, what is it that normally maintains species boundaries, right? We have dogs, we have cats. Those are two very different species that cannot interbreed. What is it that keeps the separation between them? Okay, so we're going to look at two different major categories of barriers, and they are both considered reproductive barriers, barriers to interspecies reproduction. So reproductive barriers, there are a couple of different types. There are what are called pre-zygotic barriers. So let's just review this word. What is a zygote? A zygote is a sperm that has fertilized an egg. So we've joined those two groups of chromosomes together. That results in a zygote. So pre-zygotic barriers, these are referring to barriers that prevent that fertilization even from taking place. And there are 5 different types of pre-zygotic barriers that you'll need to know about, or should know about. First on the list, we have temporal isolation. Temporal isolation refers to the fact that different species may have different breeding seasons. So if you have 2 different types of animals, say, and they happen to live in the same habitat, perhaps they just don't breed at the same time of the year. So that would be a temporal isolation. Temporal refers to time, and so this is talking about the fact that different species might breed at different times of the year. We could also have habitat isolation, and so if the 2 species don't live in the same area, then they're not going to attempt to interbreed, so that would be a pre-zygotic barrier. Next on the list, behavioral isolation. Sometimes, even if different species live in the same area, they just recognize each other as being different, and so they choose not to interbreed, even if their breeding seasons might overlap. That would be behavioral isolation. It's based on the choice. Mechanical isolation, this refers to the fact that sometimes it's not possible to get the sperm to where the egg are at. Just anatomically, the 2 different species might be incompatible with each other. And then finally, the last one, perhaps you could have 2 species that are anatomically compatible, so there's no mechanical isolation, but maybe the sperm just is not capable of fusing with the egg. There's some sort of mechanism in place that prevents a normal zygote from forming and being able to undergo normal cell division. So 5 different types of pre-zygotic barriers. Okay, the other type of reproductive barrier is a post-zygotic barrier. So let's look at some post-zygotic barriers here. Not as many types, just 3 types of post-zygotic barriers that we'll need to know about. Okay, first up, we have reduced hybrid viability. All right, so in this case, we have our 2 species. They are capable of interbreeding. A zygote does form, and that offspring is called a hybrid. Okay, so it's a cross between 2 species. The hybrid, okay, it forms, but then it's not able to reach adulthood, so it's not considered a viable hybrid. It might survive for a little while, but not all the way to the point of being able to reproduce itself. So it's reduced hybrid viability. This is something that prevents this interspecies line from continuing on for very long. Okay, another type of post-zygotic barrier is reduced hybrid fertility. And a great example of this is crossing a horse with a donkey. This results in a mule. And if you've ever heard about mules, perhaps one of the things you have heard is that they're very strong. They're excellent work animals, but they are all sterile. So you can't cross two mules together. It's just once a mule is produced, that's like going to be the end of the line. Okay, so they're all infertile, they're sterile. Okay, so the last type on our list is kind of combining these two things that we've just gone over. The last on the list is called hybrid breakdown. And in the case of hybrid breakdown, what happens is you have your two species. Perhaps they can interbreed. They produce an offspring that survives into adulthood. Maybe it's even fertile. Okay, so it may survive and reproduce with others like itself, but each subsequent generation gets a little bit weaker, and eventually this line just dies out altogether. So that's called hybrid breakdown. The hybrid can be formed, but over several generations, that line will cease to exist. So this new species doesn't survive for very long. So if we have all of these mechanisms in place for maintaining species boundaries, the next question is, what makes speciation possible then? And there are two types of speciation that I would like for you to be familiar with. The first one is called allopatric speciation. And this name allopatric, what this name means is different country. Allopatric is different countries. So this is talking about a situation where maybe you have one population, one group of individuals, they're all the same species, and there's some sort of geographic barrier that ends up separating them into two groups, multiple groups. And so this could be something like a canyon opens up. So maybe we had one population of squirrels living in an area, and then maybe an earthquake happened and opened up a canyon that split the population into two groups. And then if enough time goes by, enough generations come about, then genetic drift in one group might be different from genetic drift in another. And so eventually what we might end up with is a reproductive barrier. And if we take a squirrel from each of those groups and put them back together, they're not going to be able to interbreed anymore. So that's one type of speciation, allopatric speciation. The other type of speciation is called sympatric. You can probably guess what this name means. Sympatric means the same country. So in this case, speciation is happening all in the same place. So there's no physical separation, but somehow speciation takes place. This is something that's, I would say, more common in plants. This is something that happens in plants, and most of the time it's due to some sort of mistake during cell division, and that mistake leads to an unusual number of chromosomes being present in the cells. Good example of this is polyploidy. Polyploidy refers to many copies of chromosomes being present. So remember, with humans, we are diploid. We get a copy of chromosomes from our mom, a separate copy from dad. So 2 copies total. We say we are diploid. Polyploidy is referring to many copies of chromosomes being present. So if you've ever gone to the grocery store and bought the big grapes, they tend to be really delicious. Usually they're available in the fall. Those large grapes, they're actually a result of polyploidy. Okay, so natural grapes are much smaller. And what happens occasionally with plants is polyploidy takes place, and that essentially is creating a new species of plant. It's no longer able to interbreed with other grape plants. It is its own new unique plant. And this happens not just with grapes, this happens with a lot of different agricultural crops. This is part of how we're able to sort of end up with larger produce. Having lots of copies of chromosomes present tends to have this result in plants. It enlarges structures. Okay, so let's wrap up the chapter with a little bit about plate tectonics. So plate tectonics, the crust of the Earth, you're probably all familiar with this already. The crust of the Earth is divided up into sections, and those sections can slide against each other over time. This is what causes earthquakes. And here's an interesting map. These are the major plates that exist on the Earth, just kind of a map of their layout. So each place that there is a black solid line in this picture, that's essentially, it's a fault line. It's a spot where 2 plates are sort of butted up against each other. And this movement over time can reshape the surface of the Earth. So think back to the allopatric speciation. We know that geographical features can affect evolution of organisms because it affects who's in contact with who. So plate tectonics does sort of fit really well in this chapter. At one point in Earth's history, all of the land masses were together. So at that point in time, there was a lot more sort of intermixing of organisms. We call this Pangea, when all the land masses were together. And eventually, over time, Pangea kind of split apart, and the different masses drifted apart from each other. So now we end up having these continent-specific sorts of animals, things that seem really characteristic about particular continents. So that is it for chapter 14. Those are the main points I would like